Have you ever experienced the feeling of social isolation? I really hope you have not, but chances are you have. At the time I'm recording this talk, we're still living through one of the worst global health crises in recent history. And one of the most efficient and best ways to reduce the probability of contracting the potentially deadly coronavirus is to simply distance ourselves from others. However, this comes at a price. During the COVID-19 pandemic, the prevalence of mental health problems such as depression or anxiety increased by 25%, with women and young people being the worst affected groups. Now, I'm not claiming that this is only due to the social distancing. A lot of things came together in this pandemic, but social distancing and the social isolation we experienced with it did its part. And this is something we know from several decades of research, which clearly show that social isolation leads exactly to these kind of consequences: depression-like states, states of elevated anxiety, and an impoverishment of social interactions. Now, this is nothing that is only true for humans. And this is something that for a long time we've already known to be true also for other animals. For example, in the 1960s, there were a series of experiments performed by Harry Harlow and his lab. And one of them focused on prolonged social isolation in monkeys and the behavioral consequences. Now, when these monkeys were released from isolation, they showed signs of clear panic, clear anxiety, and the way they interacted with other animals was very odd. So they were basically not capable of interacting in a normal way. When we look back at these experiments from a modern perspective, they seem pointless and cruel. But you have to keep in mind that in the 1960s, these experiments took place in a society in which the official recommendation of psychologists to parents was to simply ignore their baby when it cried in order to enable the baby to become an independent human being. Now, these experiments in monkeys demonstrated that social isolation is detrimental also in primates. But once again, this is not only true for humans or primates. In fact, several decades of research, once again, have shown that social isolation has very similar consequences on other vertebrate species, such as dogs, rodents, birds, and fish. And whenever a biologist encounters a situation like that, where an environmental challenge such as social isolation leads to very similar behavioral adaptations, we ask ourselves the question, is it possible that there are conserved genetic modules that were just sculpted by evolution in order to enable the animals to tune their behavior in order to function properly in these kind of environments? Or, when we want to phrase it differently, are there conserved genetic modules that underlie sociality, or in this particular context, that underlie the adaptation to social isolation? This was the question I was trying to answer during my PhD, and I worked on a vertebrate species, which is zebrafish, a highly gregarious basal vertebrate that lends itself quite nicely to these kind of research questions because, first, basically all molecular biology techniques are easily feasible in this species, and second, these zebrafish have surprisingly rich social lives. The way I tried to answer this question was by applying a technique called next-generation sequencing, which is an approach in which we can not only ask what kind of genes are expressed in a tissue of interest, such as the brain, but also compare different conditions. So we can ask for every gene individually whether it is higher expressed in the case of socially isolated animals or in the case of socially reared animals. My name is Lukas Anneser, and I will talk to you about one of the ways how fish perceive other fish. This is work I did during my PhD in the lab of Erin Schuman at the Max Planck Institute for Brain Research in Frankfurt. And what I found was that there is indeed a molecular tool, a molecular thermometer, if you want, that enables the animal to perceive social changes, which is the neuropeptide Pth2. So how did we figure this out? First, let me introduce you to the general experimental paradigm. We isolated animals for different periods of time, either for 5, 8, 14 or 21 days of age. The reason for this is that between 5 and 21 days, the social behavior of the animals matures quite drastically. So we were trying to capture some of the ongoing dynamics. And what we saw, and what I show you here in this Venn diagram, is the general number of differentially expressed genes. And several hundreds of genes are affected by social isolation, but when we look at the overlap, we see that only 4 genes are consistently downregulated in isolated animals at all the different time points. One of these genes immediately catched our attention, which is PTH2, or parathyroid hormone 2. Why was that so interesting to us? PTH2 is a so-called neuropeptide, or a neuromodulator that changes the way information is processed in the brain. And the way neuropeptides do this is they're synthesized in a neuron, they're released into the extracellular space, and then they wander around in the brain until they encounter a receptor they can bind to. And by the interaction with this receptor, they change the way the neuron that expresses the receptor works. So one of the ways how we can ask how important a neuropeptide is to the brain is we can simply assess how many neurons express the receptor for the peptide. And this is something I did by looking at a single cell sequencing data set, which is what I show you here. In this plot, every individual point represents one cell. And for each of these cells, we know the expression for several thousands of genes. So we can simply ask how many of these cells express the receptor. And it turns out that basically 10%, 1/10 of the entire brain, expresses the receptor and thus is at least theoretically susceptible to modulation by PTH2. But once again, what do I mean when I say modulation? When we think about information processing in the brain, we normally conceptualize it in a sequential way. Some input is impinging on our sensory organs, and our brain is trying to get the meaning out of it, and then something is happening. Either we react on a behavior level or we form a memory. For example, right now I'm giving this talk, you're listening to me, your brain is extracting meaning from my words, and hopefully you learn something. But now consider the exact same scenario. The input will be the same. This talk is recorded after all, but now imagine you haven't eaten in 2 days. Your brain will be in a very different state, and to be honest, it won't probably even care about what I say. You will be rather preoccupied in identifying the location of the nearest fridge or supermarket to find something to eat. And I think that is a good illustration of what neuromodulation is. The same information is perceived by brains in very different states, which means very different things will happen. And in the context of social isolation, this is precisely what happens. Our brains are tuned differently in order to adapt to the state of social isolation. So clearly we wanted to understand what PTH2 is doing and how it's doing it. But the first thing we wanted to understand is what kind of neurons actually produce this peptide. At this point, we understood that 10% of all the cells in the brain express a receptor, but where is the peptide itself synthesized? We performed an immunostain, which is what I show you here. This is the brain of a zebrafish, of a larval zebrafish, and we took an antibody that recognizes specifically PTH2. What we saw was that there's only a handful of cells, between 20 and 30 neurons, that express PTH2. And these neurons are located in the thalamus, which is a bit odd because the thalamus is not normally the place where you would expect neuropeptides to be produced. But when you think about it, it makes a lot of sense because the thalamus is normally thought of as a sensory relay station. Basically, all of the input we get from the environment is relayed to the entirety of the brain through the thalamus. So the cells within the thalamus are located perfectly to get this information about changes in the environment, potential social changes in the environment, and then broadcast this information to the entire brain, ultimately tuning our behavior. So, of course, this is interesting in principle, but is a molecular system like PTH2 actually capable of keeping up with changes in the social environment? Those can happen rather suddenly and quickly. In order to understand this, we tried to analyze the temporal dynamics of PTH2 synthesis. And one of the first experiments I did was I took isolated animals and placed them into a social context. And after different periods of time, I would then ask how much PTH2 was produced. Now what I show you here in this plot is the normalized PTH2 expression strength. And I normalized it to the two different states I was comparing, with 0 indicating expression levels of PTH2 in animals that have been raised in isolation since birth, and 1 indicating the levels we find in animals that have been raised with other animals since birth. Now, when we give isolated animals the ability to interact with other animals, the following is observed. After 12 hours of interaction, we see a complete rescue of PTH2. So these animals are not distinguishable anymore from animals that have been raised with other animals since birth. After 3 hours, we see a rescue of 50%, but already after 30 minutes, we see the first significant increase. When we turn the question around and ask, how quickly is PTH2 downregulated when we take a socially reared animal and place them in isolation? It takes a bit longer, but already after 6 hours, there's no difference to be seen between animals that have been only isolated for these 6 hours and animals that have been raised in isolation since birth. So this already tells us that PTH2 is this bidirectional indicator of changes in the social environment. If there are more animals, it is activated, and if the animal is isolated, it is switched off. But in fact, it's more than that. It is a quantitative encoding device that reflects changes in a very precise manner. And the reason why I can say this so confidently is that I performed an experiment addressing precisely this question. When we raised animals in different densities, either alone or in groups of 5, groups of 20, 50, or 100, and assayed how strongly PTH2 was expressed, we saw an increase for every increase in the social density, which tells us that PTH2 is more than this bidirectional indicator of social change, but it quantitatively reflects social density, thus acting as this molecular thermometer for social density. But what does it actually mean to the animal? Apparently, with this system, it can now track changes in the environment, but what does it actually mean? How does it respond on a behavioral level? In order to understand this, I needed to use mutant animals. What I show you here is the brain of a zebrafish, and we see the expression of the RNA for PTH2, of the peptide, and we see this in two different kinds of animal, either in the wild type, where we see a lot of RNA and peptide, or in a mutant. In this mutant line, there is a particular mutation in the coding sequence for PTH2, which makes it impossible for the animal to produce the functioning peptide. So regardless of how many animals are in the vicinity of these mutants, PTH2 will not be expressed. And this is important because I showed you that social isolation not only changes the expression level of PTH2, but of several hundreds of genes. And if we only compare socially isolated with socially reared animals, it would make it very complex to decipher the individual contribution of individual genes. So in what context is PTH2 relevant? Or in what context would it be relevant to look at behavior? One of the behaviors that are really interesting is predation. What I mean with this is the following. If an individual animal encounters a predator, and this predator is hungry, that's tough luck. The predator will most likely attack and potentially eat the animal. But if the animal is part of a larger group, then the risk of the predator attacking is just as high, but the risk for the individual animal to be attacked is way lower. It's diluted. The predator may attack any member of the group. And what that means is that animals that perceive social isolation should be more tuned and more able to escape from sudden stimuli in the environment. They should basically be a bit more anxious because for them it matters more to be able to respond quickly and adequately to potentially threatening stimuli. Now this is something we can actually measure. And we can measure it by looking at something that is called the startle response, which is something we find in a lot of different species. Humans are scared of sudden noises. Zebrafish are not so different. If they encounter a sudden stimulus, such as a vibration or a noise, they will bend their bodies and try to swim quickly away from this potential threat. And this is something we can then measure. So when we look at the fraction of fish that display a startle response or an escape response in response to a vibration, and we compare mutants and wild types, we see that the mutants have an elevated propensity to escape, which is just in line with what I told you before, that socially isolated animals should be more aware of the environment and should be more eager to escape. Now, this is one of the behaviors we looked at, but this is not where we stopped. We were also trying to understand what other kinds of behavior could potentially be changed by the presence or absence of PTH2. And one of the things we found very interesting was this question whether PTH2 might be kind of a primer for social behavior. So what kind of social behaviors can you look at in zebrafish? I already told you they have a surprisingly complex and rich social life. And I will just mention two of the aspects of their social behavior. The first one is something that is called social preference. This means that zebrafish will try to minimize the distance to other fish when they encounter them. And on a higher level, this leads to something that's called collective behavior, which is also what I show you in this video. If you have a group of fish, they will align their trajectories and they will jointly explore the environment and travel from A to B. So first things first, how can we study social preference? It is possible to look at this using a chamber like this. In this chamber, we have a focal animal, and this focal animal is placed in a tank which has two adjacent compartments. To both of these compartments, the focal animal has visual access through a transparent barrier, which is indicated in blue. Now, one of the compartments is empty, the other one contains conspecifics, and we can measure the time the animal is spending in proximity to each of these compartments and use this to construct something that is called a social preference index. This is a number between -1 and 1, with 1 indicating that the animal spends 100% of the time close to the conspecifics, the other animals, and -1 indicating antisocial behavior. Now, when we start by simply placing an animal into the empty tank in order to habituate, we see that it doesn't really care about any of the compartments. It doesn't really have any preference. This changes as soon as we place other fish in one of these arms. In this context, the wild-type fish will show an elevated propensity to be close to that arm. Unfortunately for us, it was pretty much the same in mutants. End of story? Not really. This experiment we first conducted with 21-day-old animals. This is the early juvenile stage. And I already told you, between 5 and 21 days, there's a lot of maturation going on for social behavior. But it's not done at this point. There's still some development happening afterwards. And we decided to give it a shot and test precisely this behavior at a later stage of development, in the late juvenile stage, or at 56 days post-fertilization. And in this context, we found that indeed the social preference in the mutants is basically abolished. They have no preference for conspecifics anymore. And we then tried to replicate this by taking the wild-type animals and raising them in isolation, and then asking them again whether they have any social preference. And also in this context, in the isolated wild-type, there was no social preference left, which tells us that there is exactly this axis from changes in the social environment to changes on the molecular level with different transcriptional levels of PTH2, which ultimately tunes behavior. Now, with this, we knew that social preference is influenced by PTH2, but what about collective behavior? We tried to analyze this by placing 20 fish in a larger environment, which they were allowed to explore for several minutes. And we were then measuring several features of the group. So, for example, we measured how close the animals were to each other. And what we found was that if you look at the distance between the nearest neighbors, there was no difference between the wild types and the mutants. If you look at the median, the average distance between the fish, likewise, there was no difference. However, the maximum distance between the fish was increased in the case of the mutants. So how can this be? That's a bit weird. How can the nearest neighbor and the median distance be the same and the maximum distance increased? It can occur when we consider that sometimes small splinter groups break away from the shoal. The shoal is basically the group of the fish. It's just one configuration of how the animals swim together. And these splinter groups, of course, have kind of the same configuration. They have the same distance as nearest neighbors and the same median distance, but the maximum distance is clearly increased. And then we measure how many times animals break away from the main group. We see that this number is clearly elevated in the case of the mutants, which means that PTH2 mutants are less capable of maintaining a cohesive group structure. So what I showed you today is that the social environment matters. It matters on a molecular level, where socially isolated animals are characterized by decreased levels of the neuropeptide PTH2. It matters on a behavior level, where anxiety is increased in animals without PTH2. It matters on the level of social interaction, where animals that lack PTH2 do not really show any propensity to go close to other fish. And on the level of collective behavior, the absence of PTH2 leads to an impaired capability of the fish to maintain a cohesive structure as a group. In other words, what I showed you is that social contexts matter. Information is differently processed in the brain of animals that experience social isolation. And PTH2 is just one of the systems, one of the molecular levers that evolution came up with to help our brains to tune behavior in appropriate ways. The interesting thing is, the more we know about how biology constrains us in our responses to different environments, the better we are equipped to build meaningful societies that are constructed in a way that is adequate for our biological needs. Thank you for listening.